Dear colleagues, my name is Valeriya Perekatova. I am working at the Laboratory of Ultrasound and Optoacoustic Diagnostics at the Institute of Applied Physics, Nizhny Novgorod, Russia. The title of my talk is Quantification of Microvasculature Parameters in Normal and Pathological Tissues based on three-dimensional raster scan optoacoustic angiography. I'd like to say a few words about what optoacoustic imaging is. This is a biomedical technique based on the registration of ultrasonic waves excited in the tissue due to absorption of laser pulses by optical chromophores. The main blood components, oxy and deoxyhemoglobin, are the strongest absorbers in the visible wavelength range, which makes it possible to use the optoacoustic method for obtaining geographic images. Optoacoustic imaging combines benefits of optical and ultrasound imaging, resulting in high spatial resolution and optical contrast. Our group develops broadband ultrasound PVDF detectors that integrated into various systems for raster scan optoacoustic angiography. The bandwidth of our detectors can be customized to cover 3 octaves from 100 kHz to 100 MHz. More information about detectors can be found on the website photacoustics.ru and corresponding YouTube channel of our lab. The upper left picture shows a portable dual wavelength 532 or 1064 nanometer optoacoustic imaging system developed at our laboratory. Our system allows 3D visualizations with 50 microns acoustic spatial resolution at depths of up to 2 millimeters in vivo. The lower figures show images obtained at the developed microscope for human palm, rabbit ear, and mice tumor. During one experiment, the large amount of 3D data is acquired, and as a result, the vasculature images can be difficult to analyze visually. This problem is especially relevant for monitoring of angiogenesis of experimental tumors, which blood vessels tend to be smaller and more tortuous compared to the vasculature of healthy tissue. On the other hand, during monitoring of human palms, age-related changes in vessels are manifested by a decrease in vessel density, violation of branching geometry, and suppression of vascular reactivity. Development of universal approach to automated quantification of blood vessel parameters could become a versatile tool for vasculature analysis. Several studies devoted to automatic evaluation of vessel structure characteristics in different applications. For example, distinguishing vascular network of healthy and burned tissues, measuring microvasculature parameters of mice skin, and automatic evaluation of vessel structure characteristics in experimental tumors. The potential of such quantification is quite high for vessel characterization. As a result, the following aim was set: to develop the algorithms extracting quantitative vascular information from the 3D optoacoustic data in 2 different applications: non-invasive monitoring of tumor angiogenesis and non-invasive monitoring of age-related vascular alterations in human skin. The algorithm was originally developed for analysis of tumor angiogenesis by experimental images obtained at previous version of dark field optoacoustic microscope. The tumor model was subcutaneous marine colon carcinoma tumor model grown in 8 biopsy mice. Optoacoustic in vivo imaging was performed on 7 days after tumor cell inoculation. The scanning was performed within 1 square centimeter region with a step of 25 microns in both directions. Acquisition time for one 3D image was 5 minutes. The stages of the developed algorithm are shown in figures. At the first stage, Franny filtration with optimal set of parameters was applied to the reconstructed image of an experimental tumor in a mouse in order to distinguish a vasculature. The tumor area is selected manually using reconstructed optoacoustic images. The following data analysis of the tumor and our tumor area was performed assuming that all voxels in vertical scan corresponding the selected planar tumor area also belong to tumor. The rest of the imaged volume is assumed to belong to the normal tissue. The results for tumor and normal tissue are shown in separate figures. At the next step of algorithm, Franny filtrated images were binarized with a manually selected threshold and then skeletonized. A 3D binary voxel skeleton is transformed into a network graph described by nodes and links. The corresponding graphs are constructed in tumor and outside tumor separately. The following parameters are extracted based on the acquired network graph: total amount of all nodes, total amount of branching nodes, vessel density calculated as total amount of independent tree structures per volume unit, total length of all vessels, average link length, and average length of a single vessel. Three examples of the resulting graphs: in tumor area, green lines, and in surrounding normal tissues, blue lines, are superimposed on the Franny filtrated optoacoustic images for different mice. In each image, the oval-shaped tumor region can be distinguished by smaller wavy vessels as compared to those in normal tissue. Peritumoral area can also contain small vessels, providing tumor blood supply developed in course of tumor growth. Quantification of the normal tissue vessels confirms their larger scale and smaller branching level as compared to the vessels within tumor. In order to quantify this difference, 6 parameters were calculated and averaged over 8 independent images of tumors. Since tumor development is associated with angiogenesis and tumor vasculature is represented by Yana vessels, their typical size is smaller than that in normal tissue, resulting in smaller average link lengths and average vessel lengths. The smaller scale of vessels in tumor results also in larger number of graph nodes and number of branching nodes, indicating the presence of typical disordered random branching vessel trees within tumor. Analysis of the vessel density confirms the presence of a large number of newly formed vessels in tumor, which is approximately 3 times larger as compared to that for normal tissues. As compared to normal tissue, such features as small-sized vessels, their high density, tortuosity, branching and chaotic location are typical for the vascular system of most of solid tumors. An interesting fact that the total length of vessels within tumor is larger than that in norm, which is explained by high tumor demands in oxygen required for its fast development. Further, the developed algorithm was tested on the optoacoustic data obtained during in vivo non-invasive monitoring of age-related vascular features of the skin and underlying tissues of both palms. In vivo studies were conducted with 30 healthy volunteers aged from 19 to 74 years old. From 2 to 8 measurements were carried out for each volunteer. To analyze age-related changes, all volunteers were divided into 3 groups of 10 people each. These color-encoded figures show typical optoacoustic reconstructed images for healthy volunteers from each age groups. It can be seen that the first age group is characterized by small near-surface vessels, while with age, the density of the vessels decreases and there are less subsurface vessels. Optoacoustic images demonstrate the geographic capabilities of the developed portable optoacoustic microscope for visualizing both small vessels of the superficial vascular plexus of the skin in red and orange, and larger and deeply located vessels uh, showed in yellow and green color. 6 parameters were calculated from the graph and averaged over 3 age groups. A slight decrease in the average length of blood vessels with increasing age was revealed. There were no age-related differences in other parameters. The vessel density remains constant. But 3 new parameters show statistically significant differences between the 3 age groups: the total intensity over the entire image and the total intensity over the vascular mask and blood filling. All these parameters increase with age. To conclude, I'd like to tell that we worked on a novel algorithm for quantitative analysis of 3D optoacoustic images for tissue vasculature characterization. These parameters are extracted from the vasculature graph obtained as a result of skeletonization of a binarized optoacoustic image of vasculature. Proposed algorithm demonstrates statistically significant difference between tumor and normal tissue for all of extracted parameters and can be successfully employed for automated differentiation between tumor and norm. Also, the dynamics of calculated parameters can serve as numerical characteristics of tumor vasculature changes under different therapies, both in clinical and experimental oncology. Proposed algorithm can be employed for monitoring of age-related vascular alterations in human skin. We report on a slight decrease in the average length of blood vessels with increasing age and statistically significant difference between the 3 age groups in the total intensity over the entire image and blood filling. Thank you for your attention.